So I am informed that we can conduct this discussion session in Korean. So when I have listened to Dr. Lee's presentation dozens of times, and every time I think you were a really good presenter, and you delivered a lot of dense information. Of course, there were some ideas that you skipped, but all of the ideas closely came to my mind. And today there wasn't a lot to talk about ABL Bio, but in investment or biochemical industry, I have a couple of questions to ask. Among the ideas that you delivered, the platform technology, you mentioned this as a key idea. And in terms of platform technology, does it have expandability and does it have meanings? And since we are a company located in Korea, and when I interact with other overseas company, and all the companies in the past said that they have their own unique companies, and nowadays all companies say that they have their own platform. But is it really that all the companies have their own platform? If a company wants to say that that company actually has a platform, what kind of characteristics does that company have to have? And if you have some kind of technology, does that can that be called a platform and POC? To what extent do you have to have? And IRGO Biden or LBL Bio, among other companies. When a company has a platform, what kind of characteristics do you have to have?
Well, as I was listening to your answer, I think I can categorize my answer into two. For example, let's say there's a domestic company that has the platform technology and also a US company that has platform technology. If you compare the two different categories, let me go first to the US, more courses, the Seattle Genetics, Amgen Genetic, Regeneron. These companies used to have platforms. So they had platforms, and then they went through different failures, and they also went ahead with the clinical trials. And then Seattle Genetics decided to come up with the next generation technology. So one technology could treat many different applications, but I think let's say one therapy, if it is approved by one disease, one application, then I think that platform it has a scalability. However, if you think about the Korean side, let's say the companies, including myself, let's say we just hit the clinical stage, then we are now at a relatively premature stage compared to the US. We are still at a baby step. However, Korean platform companies or biotech companies, we designate a POC material, we do IND, and then we do clinic. Let's say we prove our results, then there is a chance for validation for that platform. So we can judge that based on the result.
Thank you for your answer. And you, including ABL Bio, for companies, if they take timely measures and show that they actually have platforms, and if they become secure their positions, then it will be really meaningful. And also, another question is about small molecule. So there are cases when they fail developing, and sometimes they fail because of adverse effects. And when we talk about new drugs, then there needs some efficacy. And also, people say production is the key. And when you develop a new drug, I haven't actually heard that production is the key. But for biopharmaceutical companies, you have been engaging in this field and developing new drugs. And in terms of new bio drugs, in terms of efficacy or development, any ideas?
I'm sure everyone or many of you know, but there's small molecule, large molecule, and the difference between antibody. I think explaining these differences is not that necessary because you already know this, but lead optimization is a very important concept because you have to choose a candidate and then you have to go through the profiling and then you have to come up with the final material. If you think about big pharmas, for the company that I went and worked for, there is a lead optimization process. Usually they use AI to do the lead optimization, but you have to put a lot of time to go through this lead optimization process. But when you do pilot, small molecules itself has a lot of homologous in the kinase. So when you use single kinase, there is not many selective inhibitors. As a result, multiple tyrosine kinesin are the majority. So what does that mean? IDC kinesin is all touched, but because AB's increased in the cancer therapeutics, the toxicity has to be balanced by the efficacy, and it has to offset and outweigh the toxicity. And e.g., a lot of e.g. options are, if they have manageable toxicity, even though efficacy is not that greater than toxicity, they tend to be approved. However, this antibody options you do, whether it's fully human live or whether it's humanized mouse experiment. Let's say there's lead candidate, and without going through optimization process, they just jump into the cell development. And for cell development, whether Samsung Biologics, Bionex, Wuxi, they all do this area. But I think if you think about our budget, initially you choose the target ray, and then until the first clinical, we put about 50 bill excluding labor. So, toxicity test and clinical phase 1 test, for example, 1,000 liters of batch production, that will be $3 million US dollars, and then which is 3 billion won in Korean won. So, large molecules, the downside is that the pace could be fast, but manufacturing cost is too high. As a result, people are saying, "Oh, then you have to increase the yield." That's why Whoosh or other company, they did 2 gram per liter, but now what Whoosh is doing, they generate 8 grams, 10 grams, or 5 grams. For our case, we do about 3.5 grams to 5 grams per liter. That's our average yield. And when this happens, depending on the volume of the clinic, we don't know whether one batch is enough or 2 batches are enough. If it's going to be 2 batch, then it'll be a 7 billion won. So, for a Korean biotech company, it is a lot of cost. It is a big budget, but antibodies and new synthetic medicine, because they have two different targets, you have to go one in hand. They have to be working in parallel. They have to be developed together. It's not like one side is better than the other. And when you're going to treat something inside, that'll be synthetic medicine, and antibody that should be outside the cell. That's the technology portfolio.
And when I got your business cards, there were venture capital people and business workers as well. And when we secure capital, and why the reason why I brought this question up is that you need a lot of money, and when you don't think about money and prepare, then there are cases when even though you advance to a certain point, but you cannot realize that goal because of money. So that is why I brought this question. And among the ideas that you delivered during your presentation, the costs that are linked to developing a new drug is keep increasing. And in fact, in Korea, of course, has been advancing. However, by investing trillions of won into developing a new drug is not easy. So if Korean companies keep working on this, then as time goes by, it is more difficult to raise money. So unlike the past where if you come up with a really good product, do you have to keep waiting and waiting for getting a license? And when you license, and then if you do not want to license out to other companies, then should Korean companies make a union? Should make an association? In Korea, there are raising attention in this bio industry, and how are we going to strategically come up with a new goal? And what do you think, Doctor? Your ideas?
Well, maybe this is my personal opinion, because depending on companies, CEOs have different visions and philosophies, and they have different wills, and they have different ideas. However, in my personal opinion, I think so far, as you mentioned, no matter what biotech company it is in Korea, they need to are struggling to get this funding and also a human talent. If you think about how big pharma works in the USA, when an R&D project starts, before it gets wrapped up, about 150 people get involved, from the discovery to clinical stage 1, 2, 3. I would say 150 come in and then put their hands together and then get this done. So a lot of people have to come in and join the rank. So, as I said, collaboration and open innovation is very important. And among the biospecific antibody therapy, there's IMAC, which is a Chinese company, and we did joint development. And for the last 2 years and a half, this year finally, we are going to do 2 R&Ds in the U.S. That's happening right now. And since a few months ago, we started a new project. So what does that mean is, of course, ABL cannot cover 20 billion dollars, but and also we can't have a human talent on our own. So both companies have to compensate. They have to drive these. For example, do 10 bill and 10 bill, and then you do transfer of the technology, and then we are going to do cost revenue sharing too, 50-50. And by developing ADC technology, we out-licensed this to Siston. And of course, we wanted to carry it on, but as we developed IDC, it cost twice more than what we spent for IDC, and that was just too much money. So eventually, as I said, Morphosis, Genco, just like these companies, when you go work on it over time, it can generate revenues by partnership, by from royalties, and then by doing that, you can do your in-house M&A or development. Then I think in 10 years, these strong, great companies are going to come up.
그런 방향으로 가겠다고 정했고요. 저희 말고 다른 회사들은 다른 어떤 방향을 정할 수는 있더라고요.
알겠습니다. 좀 다른 얘기를 하나 드리면 국내 바이오텍, 국내 제약사, 해외 바이오텍, 해외 제약사를 보면 항암제를 개발하는 회사가 정말 많습니다. 정말. 그래서 뭐 다른 나라에 있는 건 다 망하고 우리나라 것만 성공하는 걸 하더라도 너무 많은데
그렇죠.
그걸 보면 다 성공했으면 좋겠지만 결국은 이제 비교 위기라는 게 있을 거고 그래서 우리가 이제 투자자들이나 보통 사람들이 볼 때 어떤 회사들이 항암제를 개발하고 있는데 저거는 좀 더 의미가 있다. 저거는 좀 그런데 뭐 이런 것들. 그래서 뭐 그런 판단을 파이프라인 보고 어떻게 해야 될까요?
이제 그 항암제에 대한 그 예를 들어 성공률은 통계에서 보시면 신약 개발의 성공률이 예를 들어 저희가 9.6, 9.7%면 항암제가 5.7% 정도뿐이 안 되니까 반뿐이 안 돼요. 굉장히 어려운 건데 저는 이렇게 봐요. 그러니까 예를 들어서 제넨텍 하면 거의 브레스트 캔서, 유방암을 거의 그냥 타파했다. 그다음에 최근에 에스트로제니카는 령 캔서, 폐암을 거의 대체했다. 그리고 이제 J&J 같은 회사는 최근에 보면은 멀티플 마이엘로마를 거의 대체하고 있는 것 같아요. 그러니까 이제 외국 회사들은 나름 자기네들이 하고 있는 인디케이션에서 굉장히 포커스돼. 물론 그것만 하진 않지만 그 나름 회사에서 이거만큼은 우리가 어떤 회사가 바이오시밀러가 들어오건 신약이 들어오든 혈통같이 지킬 수 있는 강력한 파이프라인을 갖고 있다. 근데 이제 오픈된 필드에서 보시면 요즘 약이 없는 뭐 췌장암, 팬크레아틱 캔서, 프로스테이트 캔서 당연히 이런 게 중요한데 뭐 이런 회사들이 못 만드는 약은 대한민국 회사들 만들기가 어려워서 서로 굉장히 스트럭을 하고 있는 건 맞는 거예요. 이제 저희도 실은 ABL001이라는 어떤 물질을 하나 때부터 저희가 이중항체를 해서 요즘 임상 결과가 좋아서 저희가 팬크레아틱 캔서에서도 데이터가 좋고 비암에서도 좋고 뭐 괜찮은 데이터들이 나오기 시작을 해요. 근데 이 위암 같은 경우는 외국 회사랑 얘기했을 때 이 매력도가 좀 떨어지는 경우는 있는 것 같긴 해요. 그래서 이 기술 이전을 만약에 전적으로 목표를 하면 이제 웨스턴
So if you're only only focusing on technology
근데 대한민국의
to focus on indication for foreign people. But in Korea, blood cancer, we do not have that many portfolio, but there's a strong need from foreign companies for blood cancer. So you have to target unmet medical needs and then specify a target. You work on it, and maybe we're just doing a clinical 2 after clinical 1. But once you keep working on it, if you take and embark on this journey, a very tough and difficult journey by identifying indication, there will be a good result.
Thank you. If that is the case for individual companies, then like, you should take a close look to what overseas or global companies are doing. Well, then, you're working on anti-cancer, and what kind of different characteristics should we pay close attention to?
Well, if you think about our company, we do CNS, but other big pharma, if you look at their articles, as CEO changes, their application changes. For example, the current industry trend, CAS was basically buried and then it revives after CEO gets replaced, and NACI or other applications are out there. But even if there are other applications, big pharmas and US pharmaceutical companies are focusing on anti cancer and CNS. Of course, right now they're focusing on infectious diseases because mRNA is leading many different applications, and because there are so many vaccination, infectious diseases were neglected in the past, but because of this, the pandemic, it is emerging as a new trend. So, in my personal opinion, anti-cancer drug, CNS, and infectious diseases. These three are going to be key areas that are be interested by big pharmas.
Thank you. And here comes the last question, and I was kind of confused. Some people approached me last year, and since they know that I was kind of engaged in bio-specific antigen, and they say, so probably three antigen. Why not tenfold antigen? They say. But today ADC and bispecific antibody were discussed. So what kind of limitations do we have? To what extent can we extend this? And since we have limited room for labs, and if to make something even fancier, if we say tri-specific antibiotic or so, but in the future, if we take this kind of a multidisciplinary approach, what would be in the future?
Well, whether it's biospecific or ADC technology, if you follow the history of their development, you'll have an idea. When I started my company, ABL, I talked to other companies. I also talked to Hwang Yu, and then when I was doing IR for IPO 2 years later, the most frequently asked question was like, why didn't you do joint therapy? Biospecific has really low yield. It's not that effective. However, I would say that biospecific antibody, the yield, the productability is not an issue at all, because when Hanwha was developing it, it was 1.1 gram per liter. But Bionix, I'm not saying I'm looking down on them, but even Bionix, a Korean company, is yielding 3.4 gram per liter. So productability is already overcame. And as you understand biology, and if you look at all the failures of clinical trials, especially immuno-oncology drugs, biospecific treatment is better than the other option. And there has been accumulated data that proves it. That is why biospecific is considered the mainstream. But if you think about triple specific, there are companies that are claimed this. For example, whole setting or Obitoth is added, and then PD-1 is added additionally, and that is making up a three antibiotic. But there is a productability issue. Like the concept POC works, but the actual production is not at that stage yet. I think in about 5 to 10 years, three triple specific antibodies could be derived, but we have to understand the lesson. What J&J Johnson and Johnson is doing is they are trying to do a combination of biospecific antibody and another biospecific antibody, because 5 years ago, even 5 years ago, people didn't even want to mix up a single monoclonic antibody. But I think if they're now trying to combine two biospecific antibodies, I think triple specific antibodies are going to be generated in the future. When first generation fails, second generation fails, and then now people are talking about third generation. That is why NegoCan and us are creating NegoCan antibody, and we did technological transfer. And for next generation joint development that we are doing with NegoCan, our bispecific antibody is going to be added with ADC. So we're going to go for biospecific antibody ADC. That's the conclusion of our companies. We're still at the early stage. What we would like to do is when NDC is added, we want to know whether a biospecific antibody is going to prove better efficacy than the conventional ones. I think we will need about 2 years to be able to give you evidence or proof. So in that sense, doing tri-specific antibodies, okay, it's fine. But the mainstream right now is bispecific, and the ones that are prepared with this wave will be able to survive and go for the next step.
Well, if you take the real situations, we should keep working on that. Thank you for your answer.
Thank you very much for your discussion.